Okay, so in this podcast, I'm going to talk to you about microscopes. Okay, so when you look at a microscope, there's really two main things you're looking or focusing on. How zoomed in are you? So the magnification, and how big is it? Or the resolution. And that's just, you want to know that resolution is the ability to see fine detail. And so this would be if a picture or an image is blurry or not blurry. So resolution is like the opposite of blurry, so it's sharp. The resolution is the ability to see fine detail. These are inversely related. As magnification goes up, resolution goes down. And so as the magnification, if you zoom in on a wallet photo, then and make it a 16 by 20, it's going to be blurry because as you zoom in, resolution decreases. Okay, the resolving power, that's how good your microscope is. That is how close can two items be and you still be able to see that it's two items. And so if here's a dot, like a bacteria, let's try this. Okay, so there's a bacteria and there's a bacteria. And they're so close together, but you can tell that they're two dots by looking at it. And so if you have a resolving power of 2, that means if two bacteria are 2 nanometers apart, so that's times 10 to the negative 9, that's really small, then you can still tell they're two different dots. So let's just say, for example's sake, these are 2 nanometers apart, and these are 4 nanometers apart. And so the smaller the resolving power, the better your microscope is. Okay, so we did this in lab. How to calculate the total magnification would just be the ocular, which is normally 10, but can be 15 or 20, times the objective. And remember, we went over in class, you want to know that scanning is 4, like you want to know the word and the number. Low power is 10x. 40 power is, 40x is high power. And then 100 is that oil. And so when you're looking at, let's just say, low power, for example, your total magnification is 10 times 10, which would be 100. You're looking at it times 100. Okay, so 2000 is as magnified as it can get with a compound microscope, or with a light. When it says compound microscope, that's just with a light microscope. And anything higher than 2000 is going to be distorted. The image is going to be blurry, and the adequate resolution is impossible. As the magnification increases, so like as you go from low power to oil, you will need to turn up your light. And we went over that in class. You have to physically do it. Okay, the oil immersion, you put a drop of oil on your slide with that objective, and the oil oozes. I think I have a picture. The oil oozes up around. Nope. Okay, so here's your objective. Here's your objective lens, and here's your slide. Okay, so the oil, which will be yellow, oozes up around the objective lens, and it creates a closed system so that the light can come straight through. And there's all the parts. We went over that in class. Okay, when you look at a microscope, this is just where the light goes from the light bulb, the illuminator, all the way to your eyes. And so think about the microscope. It goes, I can probably do it from here. Okay, so it goes light source. So you look at your notes. Light source. And then it goes through the slide. It keeps going up. So it's going to go out this way. Right? So it goes light source. And then the diaphragm, except it's called a condenser on the notes. So that's what you want to know for your test. That little lever under the stage that allows you to adjust the light. It's going to go through the slide, through the objective, through a prism, and then to the ocular. So you want to be able to pick that out from the light source, which is the light bulb, all the way to the oculars. Okay, bright field microscopy is what we will use most of the time. And that's just where the background appears white and the bacteria are stained a color. So the specimen is dark and the background is light. Okay, so we have compound light microscopes, but there's another type of microscope called an electron microscope. And they look like this. They don't look anything like the microscopes we have. And the image you see on a computer screen. And so you put the slide in right here, and then you look with your eyes right here, and the image is on the computer screen. So this is going to magnify much larger. So if you go back, you can see these are going to use electrons, not lights. Hence the name electron microscope and light microscope. And so this specimen appear on the screen. This made it possible to see really small things. So they're really expensive, and you can't, with the electron microscope, all of your slides have to be pre-prepared in order to go into the computer. And so you can't see anything living. So you couldn't ever see how a bacteria moves or how a bacteria grows and divides or anything like that. But it is really neat because you could see, inside the bacteria. So there's two types of electron microscopes. There's transmission, which really lets you see the surface of the organism. I guess it, looks transmittance across the surface. And then there's scanning, which shows like a 3D image. And so if you look to the right, that's probably E. coli. And you can see it looks 3D. So that's with the scanning. And there's an ant with a scanning electron microscope. I just thought that was neat. Okay, so with staining, we're going to stain the organism. And so that this sounds difficult, that paragraph does, and it's really not that difficult. When we stain the bacteria, what we're doing is we're making it so you can see the bacteria, right? Well, the reason you can see it is because light passes through the clear slide and the unstained bacteria at the same time, so you wouldn't be able to see it. And so by staining it, we're slowing the light down so you can actually see, what color it is. Okay, so staining is done by applying dyes to the specimens. Dyes are salts composed of positively charged ions. Anything that's charged is an ion, but a positively charged ion is a cation. And see how it has a T in it? That's easy to remember. That's your positive. Cation is positive. Anion has an N in it, and it's negative. And so the dyes are positive and negatively charged. In each dye, it's going to, either the positive or the negative is going to attract and bond to the organism, the bacteria, to make it dye. And so basic dyes are positive or cations. Acetic dyes are the negative ones, and they are the anions. Well, bacteria have a negative surface, and so what kind of stain would you want to use? You'd want to use a basic stain because, positive and negative are going to attract. I'm going to give you a clue. All the stains on the first exam, because there's a lot of questions. And so all the stains on this test are basic or positive. And like I said, there's a lot of questions on this test from the microscope, so make sure you pay attention to this PowerPoint. Okay. The basic dye is for the bacteria, and so we went over that. Acetic dyes are used when the bacteria won't stain, and so acetic dyes are used to stain the background. And so it would look like this. So we'd stain the background pink, and then the bacteria would be white. And so that's not what you saw in class. Yours was opposite. The background was white. So an acetic dye is when the background is dyed. And a lot of times that's used for a capsule stain because a capsule won't stain. And so you put the stain on the slide and it stains everything but the capsule. Okay, so in class, when we do the gram staining, you have to first make a smear. And you're going to take a small scoop of bacteria and put it in a drop of water. And then you're going to make a little puddle of bacteria water. And you want your puddle to be about the size of a quarter. And then it's going to air dry. Okay, I'm going to tell you in class too. The bigger your drop of water, the longer you have to wait for it to air dry. And so if you want to go home that night, which we all do, you're going to make that drop of water tiny, tiny, tiny. So that when you smear it out, it takes 10 minutes to dry, not 45. Okay, then you're going to fix the smear. And all that means is after you've made it and it's dry, put it through the flame, go through the flame once and then right back through it. And that's it. You don't want to hold it. And if you do hold it, you're going to charcoal it. And then you'll see brown when you look at it. But the reason you fix it is two things. It's going to kill the bacteria, even though it was so fast. 1, 2. And it's also going to make the bacteria stick to the glass. So fixing the bacteria kills it and makes it adhere to the glass. Okay, so in microbiology, we've got 3 different kinds. We've got simple stains, which is just, and that's what's on this slide, so I'll go ahead and talk about it, which is just coloring the bacteria. You can see, is it there? You can see the shape of it, the morphology of it. And you could see, the count, basically, the presence and the morphology. And so you're just going to do basic dye, wait 60 seconds, and then, you'll be able to tell. When I first started teaching, I was teaching a high school class, and we were going to look at some of their cheek cells out of their mouth. And I needed normally we use methylene blue for a simple stain. And I didn't have any. And so I thought maybe I'll use food coloring and maybe it'll work. If it doesn't work, oh well. And it worked perfectly. So a simple stain is kind of like that. You're just coloring it so that you can see if it's there. A differential stain, you're going to be telling a difference in something. And the one we're going to do in class is the bacteria gram-positive or gram-negative. And then a structural or special stain, that's going to let you see, like, does it have flagella? Does it have cilia? What kind of structures does it have? Okay, so the differential stain, we're going to focus on the gram stain in lab, but the acid-fast stain is a good one too. So we're going to talk about both of those. Okay, with a gram stain, it divides bacteria up into 2 main groups. And those 2 main groups are gram-positive or gram-negative. And so if an organism is gram-positive, it's going to have a thick peptidoglycan layer. And if an organism is gram-negative, it's going to have a thin peptidoglycan layer. Okay, so here's your gram-positive. And here is your gram-negative. Okay, so gram-positive we said has thick peptidoglycan. While gram-negative has thin peptidoglycan. Okay, and the gram-negative also has some stuff on the outside. It has basically a cell membrane on the outside. So it has a polyliposaccharide layer. And so you want to know that. And the gram-positive has the peptidoglycan that's really thick. Okay, so I'm going to walk through the gram staining procedure with you. With this table, I'm going to draw it. And you follow along in your notes. Okay, so here's your cell. Step 1. Okay, so the first thing is you add a crystal violet dye. And this is a basic dye. This is, if you're looking at your notes, this is a primary stain basic dye. And so you put it all over the bacteria. So that's step 1, and that's on gram-positive and gram-negative. I'm just putting it over here on gram-negative. Step 1, crystal violet. Okay, you want to know that that's basic stain, primary stain. Remember I told you all the stains are basic stains, which means they have a positive charge. But this one is your primary stain, is the first one. Okay, the next step is iodine. And iodine is brown, because iodine is brown. And it's not a dye. It's what we call a mordant. And so the iodine binds to the crystal violet, and mordants, if that's a word, it binds to it like that. It makes the crystal violet a big molecule. And so what it does is the crystal violet and the iodine bind together now, and they're trapped inside the bacteria. Okay, the next step, before we go further actually, let's make our peptidoglycan cell wall over here really thick. And the other side is really thin. Okay, step 3, you're going to do, I guess black will work. Okay, step 3, you're going to do, it's a decolorizer. It's basically fingernail polish remover and an alcohol mixed. So you want to know what it's made of. Decolorizer. Okay, so the decolorizer does just that. It removes the color. Okay, well, this over here is really thick. And so it can't, oh, that was a little much. It can't do that to the gram-negative side, see? I mean the gram-positive side. The decolorizer kind of just does like that. It doesn't do anything. The gram-negative, on the other hand, it's going to kind of, see how I made a hole in the peptidoglycan? So that the crystal violet and the iodine all come out. So after the decolorizer, gram-positive is purple or violet in color. Right? That's why the colors were written that way. Gram-negative is clear, or colorless, I guess is a better word. And so after the decolorizer, you want to know what color they both are. Gram-positive is purple. Gram-negative is clear, purple or violet. Okay, what if we had looked at them after the iodine? What color would they both be? They'd both be purple, right? Okay, next, step 4, you're going to add saffron red. And I guess I can make that red, huh? Okay, and that fourth one, when you add the saffron red, it's going to go on both sides, but it's going to make the gram-negative actually a pink or red color. The gram-positive, which was already purple, it really just made it a darker purple. And so the saffron red, you want to know, is also a basic dye. It's the counter stain. So the only reason you do the saffron red is to dye the organisms that are gram-negative. So at the end of this staining procedure, when you look at bacteria under a microscope, you'll be able to tell whether the bacteria is gram-positive, purple or violet colored, or gram-negative, pink or red colored. And so that's the whole point here, is to see which type of cell you're looking at. And then we treat those two types of bacteria differently. Gram-positive, we're going to target the thick cell wall. Gram-negative, we're going to target that lipopolysaccharide layer. And so you want to know these steps, what the different names for them are. So crystal violet is the primary stain. It's a basic stain. Iodine isn't a stain. It's a mordant. Decolorizer, that's alcohol and acetone. And then saffron red is a basic stain. Okay, then also at the end of this PowerPoint, there's some questions. Think about them. What if you left the iodine out? Then both would be red probably, because the iodine makes that crystal violet molecule big so that it doesn't come out of the gram-positive. What if you left the decolorizer out? They'd both be purple. What if you left the saffron red out? Well, the gram-negative would be clear or colorless. Okay, so hopefully that helps. And all this does is it goes through what I just said. But I just thought it helped to draw it out. And so that's going to be the gram-positive. That's all going to be the gram-negative. And then take a minute and consider the answer to these questions. So pause the video, try to answer them, and then when I answer them, you'll see if you got it right. Okay. So the purpose of the mordant is to bind to the crystal violet to make a big molecule. Crystal violet is basic. Saffron red is basic. Graham's iodine is not a dye. So that was a trick question. In each case, how do you know? Because they stain the bacteria. And because I told you. Okay, so just talking about gram-positive. After the primary stain, it's purple. After the mordant, it's purple. After the decolorizer, it's purple. And after the counter stain, it's still purple. So gram-positive is going to be purple or violet in color. Gram-negative, after primary stain, is purple. After the mordant, it's purple. After the decolorizer, it's colorless. And then after the counter stain, it's red. If the mordant was omitted, then the crystal violet would come out of both sides, and they'd both appear red. If the decolorizer was omitted, then the crystal violet wouldn't come out of either, and they'd both be purple. Okay, and then an acid-fast stain. Some bacteria on the outside, specifically tuberculosis, so Nocardia and tuberculosis both, but they have an outside layer made out of wax. And this wax is called mycolic acid. And so an acid-fast stain is doing just that. And I want to focus on, the important things here. The acid-fast stain dyes the mycolic acid pink, bright pink or red. And so if the bacteria has the acid-fast, if it has the mycolic acid, then it's red. And if it doesn't, it's blue. And so this was a good test for tuberculosis, because tuberculosis, Mycobacterium is tuberculosis, by the way. It's leprosy too, but that's the genre. of tuberculosis. But, if the scratch test, it gives a false positive. And so a lot of times they'll do an acid fast stain just to make sure that it's tuberculosis. And so I hope this was helpful.